Okay, good morning, everybody. First, I would like to thank the Education Committee for giving the opportunity to give you an update of what we call now TP53-related cancer, because as you know, LFS was considered in the old day as a very rare disease, and we are dealing now with not a very frequent disease, but nevertheless a genetic predisposition remarkably characterized by a wide tumor spectrum and diversity of severity. Just to introduce my talk, and to fully respect Joseph Roumeny and Frédéric Lee, as you know, this syndrome was defined on an epidemiological basis based on the recruitment of probants having a sarcoma and having an excess of first or second degree relatives that correspond to the historical definition. The second step was definitely the molecular definition, like in many monogenic disorders. Two groups, one was directed by Stephen Friend, and the postdoc was at this time David Melkin, provided evidence that LFS was the result of germline p53 mutation, and as you know, p53 is the most well-known genes in human beings, the most analyzed, is the reason why there is a specific focus today on the syndrome. So, as you know, one probably the key role, and the idea is to summarize only what we know about p53 in terms of clinical application, and to follow the next slide, we can summarize the key role of p53 according to the definition of David Lane, provided in the late 80s. p53 can be defined as the guardian of the genome and anti-oncogene. Indeed, as soon as you are going to have within a cell DNA damage occurring directly or indirectly, subsequently, for instance, after oncogene activations, you are going to observe an increase of the quantity of p53 due to stabilization, resulting from the disruption of the interaction with the key regulator MDM2. And p53 is a very powerful transcriptional factor able to upregulate numerous genes involved in the main pathway, which are going to be disrupted in cancer, corresponding, of course, to cell cycle arrest, to DNA repair, and to apoptosis. And you have like a very chronobiological process, because thanks to different types of affinity, the four genes which are going to be upregulated are going to be the cell cycle in order to allow the cell to repair, and if the repair is impossible, you will induce apoptosis. So what you have to keep in mind is that the key role is a transcriptional response to DNA damage. Now, as you know, and as I say to students or colleagues, it's a disaster to summarize a life with a slide, because you have here, like for the other presentations, slides corresponding to patients, real patients, and of course, LFS was characterized in the old day as one of the most severe genetic predisposition, characterized not only by a wide spectrum, but as you know, by a very early age of onset. And you have a typical here LFS family with the index case who developed a breast cancer at 25 years of age, unfortunately lost her daughter from a medulloblastoma at 2 years, and her sister previously died from an astrocytoma. One of the second characteristics that you are going to find, of course, in all the Mendelian predisposition to cancer, but which is remarkable high in LFS, is the number of multiple primary cancer. We are not considering here metastatic evolutions. We have secondary tumors, but we have multiple primary cancer. And as you will see in the next slide, we have a huge fraction of TP53 mutation carriers who are going to develop multiple primary cancer, as illustrated by these index cases who developed the sarcoma at 2 years, and adrenocortical carcinoma at 2 years, osteosarcoma at 14 years, and left and right breast cancer at 22 and 25 years of age. And of course, when you are seeing this type of family, it's a real disaster in the family. And in contrast to other types of Mendelian predispositions, such as those related to BRCA1 or BRCA2 or Lynch syndrome, the pedigrees are moving very fast, because you have in some family at least high penetrance of the mutations. One of the significant contributions of the French group was to try over the last 20 years was to update and to provide some criteria in order to cover all the situation, because the first challenge from the clinical point of view is to suspect LFS. And so we have designed what we call the CHOMPRET criteria. Agnès CHOMPRET was a very friend, and she died, she passed away from breast cancer. So in memoriam, we designed that the CHOMPRET criteria. And the idea is to have practical criteria that can be used on a daily basis in your clinics. What is very important to consider is you have 4 boxes. Either, and we did that and we published that in the Journal of Clinical Oncology 3 years ago, and we have sequentially updated this criteria. Either you have what we call the familial presentation. By familial presentation, we mean that you have a proband and a first or second degree relative, because you have to integrate the incomplete penetrance, and both relatives should develop what we call a LFS tumor, corresponding to very early breast cancer, only in female, soft tissue sarcoma, all types, osteosarcoma, CNS tumor, and adrenocortical carcinoma, and we put a cutoff in terms of age, before age 46 in the index case, and for the relative 56. That's one possibility. But what is very important, and that very important message, is that the familial history is not mandatory. So you can have another solution corresponding to multiple primary tumors in a single individual, developing, of course, at least 2 LFS tumor spectrum, the first of which occurring before 46 years. Or you are going to have the representation of rare tumor, which are very suggestive of LFS, such as, especially in the childhood, adrenocortical carcinoma, choroid plexus tumor, rhabdomyosarcoma of embryonal anaplastic subtypes, some types of medulloblastoma, irrespective of the family, and more importantly, what we can call very early breast cancer, as defined by breast cancer occurring before 31 years of age. And just to convince you that the familial history is not mandatory, we estimate the mutation detection rate of germline TP53 mutation based on the single criteria, without additional familial criteria. And as you can notice, we have a very high mutation detection rate, roughly 40%, for instance, in children, especially with ACC, with children with choroid plexus tumors. And notice that if you are considering only early onset breast cancer, you have a mutation detection rate of 6%. As you know, the mutation detection rate of BRCA1, BRCA2, and even if you include PALB2 in Europe is today 9.9. So in other words, 6% is really significant. So just to give you, it's not an estimate of the penetrance, because of course the question of the penetrance is a burning question. It's like a global picture. From the practical point of view, you can consider that you have 2 parts in the LFS. You have the childhood part and the adult part. And in children, you have a wide spectrum characterized by osteosarcoma, adrenocortical carcinoma, CNS tumor, soft tissue sarcoma, and you have here the percentage of affected mutation carriers that we have in our French cohort. And as you can see here in adults, of course, the spectrum is more narrow, and you have 2 emblematic tumors corresponding to female breast carcinoma and soft tissue sarcoma. The question of breast, I put that on the table because there is a huge controversy, and of course we have probably opposite view. Nevertheless, I think that we are reaching now consensus, and we had many meetings in Toronto, in Chicago during the SCR, because, you know, today people want to put p53 in all the panel genes. And it's a very burning question. And I would like to highlight that we don't think it's a good idea. And I would like to give you some argument for that. So we did a first study several years ago, and we analyzed systematically p53 on the criteria of very early breast cancer. And we took that without knowing the familial history. And we have 14% in terms of mutation detection. And as I indicated on the previous slide, when we restrict that to sporadic cases without additional criteria, the familial level we had 6%. So we have a huge rate here. Do we have some specificity as the Dana-Farber group published several years ago? We have probably a high percentage of RBB2 amplification samples, what we call the triple positives in terms of RB amplifications, progesterone and estrogen receptor expression. So you have the mirror pictures of BRCA1. Now, we colleagues perform a huge study in France on more than 5,000 families suspected to present an hereditary form of breast cancer. And we try to very curate the data. And if you just put on a panel P53 without respecting the criteria I indicated, as you can see, we have a very, very low mutation detection rate. In our study, it was even less than 1%. The second notion, which is also now a common question in the NGS area, like in other Mendelian disease, is the contribution of de novo, since we hadn't estimated the de novo mutation, as you know, and we have a good estimate thanks to the trio analysis. We try to estimate that. So we restrict the analysis on more than 300 cases, and we systematically perform target sequencing. We check, of course, the paternity. And basically, we were able to demonstrate that on this theory, we had at least 40 de novo mutations, indicating that the rate of de novo mutation or the contribution of de novo mutation to LFS was at least, because we have probably estimated that, 12%. We check also using NGS that the parents themselves were not a mosaic. So we had here real de novo mutation corresponding to classical pre-zygotic events occurring in the germ cell. Now, the type of de novo mutation corresponding to a challenge at the technical level and revealed now by NGS technology correspond to MOSAIC. As you know, MOSAIC corresponds to post-zygotic mutation, and for clinicians, it's absolutely critical because, of course, you miss usually this type of mutation starting from blood. And the consequences in terms of genetic counseling are absolutely important, as you know, because you have no risk for the SIPs, but probably for the son or daughters. We had already evidence that MOSAIC exists because we had even using old techniques such as Sanger sequencing, for instance, in this child who developed an adrenocortical carcinoma at 4 months of age. You can see this tiny peak on Sanger sequencing suggesting the de novo mutation, which was confirmed by next generation sequencing performed at high depth. We had already another example of a child presenting a medulloblastoma. And when you are looking at that, it was very interesting because if you are looking at the result we got, in the index cases, we had here a perfect heterozygous mutations. But the question of the MOSAIC was not in the index case, was in the father, because if you look very carefully, you can see the peak. And in fact, using different techniques before NGS snapshot, we were able to show that, in fact, the mutation was present as a MOSAIC in the father, so in the germline, and with, as you can expect, the consequence. So we can have, of course, a real germline mosaicism. So in order to have a more systematic estimation, we decide to analyze systematically by next generation sequencing, perform high depth, using different algorithms, focus on detection of MOSAIC. We decide to reanalyze more than 100 cases, strongly suggestive of LFS, but without detectable alteration, using classical methods, Sanger sequencing and multiplex techniques for genomic rearrangements. And here you have the results. So we took, of course, a very suggestive situation, children with adrenocortical carcinoma, choroid plexus tumors, female with very early breast cancer or multiple primary cancer, corresponding to LFS spectrum. And as we see so far, and more, we are detecting MOSAIC. So using this systematic approach, we detect 7 additional cases. We have 9 cases in total. So one taking-home message is probably that at least 10% of the clinical situation strongly suggestive of germline P53 mutation correspond to MOSAIC alteration. In other words, when you have a situation strongly suggestive as ACC, CPT, and early breast cancer or multiple primary cancer, you should perform NGS at a high depth in order to detect the MOSAIC alteration. So you have this type of example. You have here, for instance, this bilateral breast cancer 2734, without, of course, any detectable BRCA1, BRCA2, PALB2, and P53. And when we perform NGS sequencing, as you can see, we have a mutation in 70% of the reads. And in another example, for instance, in a girl who presented an emblematic tumors corresponding to adrenocortical carcinoma, we are able to reveal also, thanks to NGS, a stop alteration within the TP53 gene. Now, what is extremely important, and I would like to highlight that, it's not because you detect a P53 mutation in the blood that corresponds to a MOSAIC, you should check that. And of course, what is extremely interesting is to analyze systematically the tumor sample, because of course, you can use the malignant cells and you can use the non-malignant cell derived from the tumors. And what is interesting is that, for instance, of this different example, you can, of course, clearly see the P corresponding to the mutant and the famous, according to the Knudsen hypothesis, detection of loss of heterozygosity, which you are going to observe in most of the cases, which correspond to the complete inactivation. So you have a demonstration of the causality of the P53 in the appearance of the tumors. But please, please, please be aware, not all P53 mutations detected in blood correspond to mosaic alterations. That was the last case sent by colleagues in France. They decided, I was against that, but they decided to include systematically P53 in a large panel. And in this aggregation of very late cancer, so in these cases, developed a metastatic bilateral high-grade serous ovary carcinoma at 70 years of age, and her mother died from a gastric cancer. They identified what they thought was, of course, a mosaic alteration, because they identified a missense mutations considered as a deleterious, class 5, in 23% of the reads. But I would like to highlight that the frequency of somatic P53 alteration in serous ovary cancer is more than 90%. And as you can imagine, it was obviously a somatic mutation. So you were dealing with circulating DNA, and it was a somatic alteration corresponding to the metastatic situation of the patient and not of the germline, like in the mosaic states. The second problem that you can have is what we call clonal hematopoiesis, especially in old patients. Sometimes you are going to see in old patients this type of mosaic alterations, but this type of mutation of alteration will precede the development of a leukemia. And you have to be aware of the situation in terms of treatment, but we know that now some treatment, some genotoxic chemotherapy, are going to induce P53. So it's very important to check the reality of this alteration. Like in all the clinical, in the genetic situation, you should always interpret your results by considering the phenotype. The first rule is first, consider the clinical presentation. You have early breast cancer, it's strongly suggestive. You have this type of situation, it's not suggestive. And check the presence of the mutation on the sample, such as, for instance, buccal swabs. So just to move to the next chapter, what you have to consider is that, of course, de novo mutation play a key role, pre-zygotic, post-zygotic. It's the reason why familial history is not mandatory, and you have also another factor corresponding to incomplete penetrance. And you can consider that 10% of the situations strongly suggestive correspond to mosaic alteration. So therefore, probably the real contribution, if you had pre-zygotic and post-zygotic of de novo alteration to LFS, is at least 14%. So now I would like to move to some burning question, an extremely burning question which was discussed during the last ACR meeting in Chicago. Probably we should today, in 2018, completely have a new lecture of what we can call the penetrance. Because we thought that, of course, the penetrance of germline P53 mutation was very high. It's not true. First, as clinicians, we are absolutely innocent. Our perception of all disease, of penetrance, is biased because we have, of course, we are seeing patients, not healthy subjects. So we are suffering what we call from a preferential ascertainment. As I just highlighted, familial history is not mandatory. I indicated that if, of course, the absence of familial history can correspond to a germline mutation, that can be explained by de novo, but also by an incomplete mutation, by incomplete penetrance. But more importantly, and that's very important, especially to the young generation, it's not because you are detecting even a class 5 alteration using NGS that it is a causal alteration. And that's probably the danger of what I can call the slippery slope of NGS. There is a risk today that we are going to decrease the quality of service because everybody playing NGS, putting mutations, and the time to interpret the variation is now very important. Indeed, other NIH group showed recently the prevalence of class 4, class 5 p53 mutation in the general population, that was the exact population, is 0.2%, so 10 times higher than the previously estimate. If you are looking at the famous example of what we call the founder Brazilian mutation, corresponding to this ARG337 histidine, it's 1 among 350 newborns. And recently, the Dana-Farber groups published a large paper based on a very large number of samples analyzed by Ambry and myriad companies, more than 40,000 samples, including 20,000 breast cancer unselected, and the mutation detection rate was 0.2%, so in agreement with the publication of our group, and you see that it's only the same that in the general populations. The second thing, and that's also a moving question, not only for p53, is that we know that germline p53 mutation have not only an incomplete, but a variable penetrance. So we had this black and white story. Now we should consider two parameters in practical genetics. First, the classification, class 5, class 4, and second, penetrance. Both items are different items. Indeed, we had some arguments when we dichotomize the presentation of family based on the French cohort, considering the type of alteration. If you look at the bottom of the table, you can see that the dominant negative missense mutations, correspond to missense mutations, which have been shown to produce a mutant protein able to trap the wild type and to inactivate the transcriptional activity. And as you can see, we have a significant earlier age onset as compared to other type of alteration, especially with what we call the loss of function mutation or the insertion, frame shift, splicing, stop alteration, supposed to just inactivate the protein. And more importantly, if you take the extreme form of null mutation corresponding to genomic arrangement, you have a huge. So we had a first evidence. And for instance, you have here the typical LFS situation corresponding to a severe form, corresponding to a missense mutation having a dominant activity. But now, if you are looking at that, it's not the same situation for the patient, for the clinician. You have here a family characterized, of course, by a Mendelian predisposition to cancer, but by late onset in the sense that you have no childhood tumors, and you have here a pure null mutation corresponding to a genomic alteration. If you are looking at that, you see that you have a pure adult form, and you have here a splicing mutation which was predicted to result into a loss of function. Look at this example. You have a stop alteration, and you have only, if you allow me, multiple primary cancer occurring in adults. So, of course, the type of alteration is not the only factor. We know now that we have modifier genes, but it's a key part of the explanation. So my group over the last 2 years tried to explore the molecular basis of the variable penetrance of germline alteration, and we decided to set up a functional assay based on the transcriptional activity of 53. The assay is very simple. First, you start from the lymphocytes. So you take the lymphocyte of the patients. You are not dealing. So either we take fresh lymphocyte or we immortalize using EBV. After that, you expose the lymphocyte, and we are using different DNA damage agents such as doxorubicin, which is commonly used in chemotherapy. And the idea is to quantify the transcriptional response. So first we did that using transcriptome analysis. Then to implement that on a routine basis in our clinical department, we select different target genes in order to have a robust test. And the idea is here you have, as indicated, the 8 of the peak correspond to the level of induction. Here you have the peaks corresponding to the level of expression before the exposure, and after the exposure. And after that, so you are starting from the same sample, expose and expose, you just quantify the level of induction, and we define what we call a functionally score corresponding to the mean of the induction level of the 6 p53 target genes we select from transcriptome analysis. And the picture is very nice, and we did that on more than 100 samples now. Look at the result we got, for instance, with wild-type lymphocyte. So you have here in blue unexposed lymphocyte in red, and you have a score of roughly 14. Now, if you are looking at a viral null mutation, you have here a complete deletion, you can see that the score is 7. And you have here the definition of an haplodeficiency. It's a real endophenotype. You are in the lymphocyte of the patient. You are not in the tumor cell line. But remarkably, when you are doing the same experiment with dominant negative missense mutation, you can visualize what we call a dominant negative activity, because as you see, you have a drastic decrease of the score, like 2. And it's a real endophenotype because you are once again not in the tumor of the samples, you are here in the lymphocyte of the patient before the occurrence of the tumors. So now we are able to classify, and looking at the different functional score, we can dichotomize between null mutations and dominant negative missense mutations. We went a little bit further and we performed ChIP-seq because, of course, the transcriptional activity depends on the ability of p53 to bind to DNA. And by quantifying at the genome scale the ability of the mutant to bind the target regions, we were able to demonstrate clearly that, for instance, as compared to the wild type, you have a huge decrease of the binding score in lymphocyte harboring only a single heterozygote mutations. And as you can see, you are in between situation for the mutations. So in other words, you can understand the diversity of the severity of the clinical presentation because the different type of alteration have a different biological impact. And just to summarize this part, we can consider also, we have to be careful, it's not the only explanation, but we have the less severe alteration corresponding to a lower penetrance, correspond to loss of function mutations. They are associated with usually later tumor onset. They are predominantly detected in pedigree with late onset cancer. Of course, the worst, as you have seen, correspond to the more severe mutation with a high penetrance, correspond to dominant negative missense mutation with the recurrent alterations. They are associated with earlier tumor onset, and there is a prevalent germline alteration in childhood cancer, explaining why you are going to find those. And you have an in-between situation corresponding to non-dominant negative missense mutation that now we call like almost lower penetrance mutations. And you have two remarkable examples of these two types of mutation, the Brazilian mutation, and we have an equivalent in France. This type of mutation specifically induce a high risk for ACC and late onset cancer. Now, in the last part of my talk, I would like to come back to the question of the multiple primary tumors in LFS. That's a very important question, as highlighted in one of my first slides. Also, this value is probably underestimated. We knew that in our cohort, at least 43% of the mutation carriers are going to develop multiple primary cancer. Remarkably, 30% of the mutation carriers who receive the radiotherapy for the treatment of the first tumors develop secondary tumors. But we have not followed all the patients, and this estimate has been confirmed recently by the NIH group. And the simple idea that you can have, which is very difficult for health professionals and even more difficult to hear for the families, is that considering the key role of p53 as a key player in response to transcriptional damage, you can speculate that as soon as you're going to expose to radiotherapy, to chemotherapy, you are going to induce, of course, you are going to recruit and you are going to stimulate the p53 transcriptional response, but if there is a defect, you might probably indirectly contribute to the malignant transformation. So we decide to demonstrate that, in fact, that chemo and radiotherapy were playing a key role in the p53 mutation carriers. So the first step was to do that in vitro. And the way we did that, we decided to inverse the functional assay I showed you. The idea now is not to start from lymphocyte harboring a p53 mutation, but normal lymphocyte with a wild type. You are using that as a living tube. And you are going to expose two different agents, either chemical agents that you want to test or physical agent. Why? Because you know that P53 is a universal sensor. As soon as you have a DNA damage of all types, you are going to stimulate P53 activity. So the idea is to quantify that and to be sure that you are looking at a P53-mediated transcriptional response to DNA damage. You are going to use this time, for instance, LFS lymphocyte as a control, as a negative control. And this time, we are not considering the functionality score, but we are dealing a genotoxicity score corresponding to average of induction rate. And we did a large work, and the work was performed in different species. First, we did that in humans. So you have the different class, you have the X-ray, you have the different classical chemo that you know, commonly used in LFS patients. You have here the value of the genotoxic score, and you can see that if you are using sodium chloride, you are going to have a 1 because you have no induction. That's your background. And you can check here the specificity with the LFS wild type. And the technical message is that, unfortunately, all the chemotherapy used as a classical chemotherapy used in LFS patients, except the mitotic spindle poisons, such as paclitaxel, are strongly genotoxic. That's the very important part. They are all genotoxic, except the mitotic spindle poisons. We decide to do that after that in mice, because it was so important in terms of conclusion. So the first thing was to do the same experiments. So we took splenocytes, roughly corresponding to lymphocytes, and we did the same experiments, and we used different target genes. And as you can see, we have the same result. So you have the human experiment, you have here the mice experiment. But we move a little bit more closer to be more than to the in vivo stuff. What we did, we took mice, and we inject or expose to different genotoxic agents. And after that, we sacrifice the mice, we extract the lymphocyte, and we perform the score. And you can see that in vivo, we got the same result, and you are now able to detect some agents which are going to require metabolism, such as here, as you can see, cyclophosphamide. We had such an evidence that we decided to demonstrate using the final proof, and we performed a 2-year study, more than 200 mice, corresponding to mice model. Since the basic consequence of all types of alteration of germline P53 is inactivations, we decide to construct plus-minus mice that we submitted to negative control, sodium chloride, chemotherapy that we dichotomize using the functional assay as genotoxic, etoposide, poisons of topoisomerase, and non-genotoxic, so the docetaxel, and X-ray. And we try to work in clinical condition. We perform monthly total body MRI and systematically autopsy. And this slide summarizes more than 2 years of work. Please have a look on this part, because here you are the situation of our patients having only a single alteration. If you are looking with a curve, so here what you are looking is the survival probability of mice and who are going to die from cancer. Since you have, of course, minus plus, you are going a spontaneous appearance, of course, of cancer. But what is remarkable, if you are looking the X-ray curve and etoposide, you have a very drastic acceleration of tumor development in irradiated LFS mice and in LFS mice exposed to genotoxic agents. So this paper is now in press. So in conclusion, we had already a very strong clinical argument, high incidence of multiple primary tumors. We had the sequential development in radiotherapy fields. We knew from the functional assays that most chemo and radiotherapy had a genotoxic effect. We have now the final demonstration in mice. So we can say, unfortunately, that chemotherapy, genotoxic chemotherapy, and radiotherapy contribute to the development of multiple primary tumors in LFS patients. So in other words, for the geneticist, we can claim that the main modifier factor in germline P53 mutation is the use of chemotherapy and radiotherapy. So therefore, to reduce the risk of multiple primary cancer in mutation carriers, if it's possible, the first rule is, of course, to treat the cancer. But if you can avoid radiotherapy, for instance, in a non-negative breast cancer, you should go to surgery, no radiotherapy, if it's possible. Surgical treatment should be prioritized, and in the future, we should push the scientific and medical and also pharmaceutical company to consider non-genotoxic treatments, mitotic spinal checkpoint if it's possible, but more importantly, targeted therapy, and more importantly, immunotherapy. So I will finish in the last 5 minutes to the burning question of the screening protocol. As you know, the SCR published the guidelines, what we call the Toronto protocol, because it was a second version of the paper published 1 year before in the Lancet Oncology. And our colleagues, and we can understand, consider 1 year ago, but it's not the situation today, that it was very important to ensure a very efficient. So for ACC ultrasound, starting from the first year of age, brain tumor, annual brain MRI, soft tissue and bone sarcoma, annual whole body MRI, which is extraordinarily heavy. As a clinician, as you can imagine, if you are a parent of the child, is very difficult. And of course, you have the same type of adult surveillance, mostly based on whole body MRI, annual brain MRI, plus in female, annual breast MRI, because as you know, the ability of breast MRIs is today, at least today, more efficient than whole body MRI to detect very early breast cancer. What are we today? We had a discussion 1 month ago in Toronto with the LFS patient associations, with colleagues from North America and Canada. We had this discussion as RCR, and we have now many papers that have clearly demonstrated using pilot studies in Canada, in US, in different European countries. Yes, you have an efficiency of whole body MRI. You have at least a 10% of cancer detection rate per year. The problem is that we have a high level of incidental MRI finding, which is of course stressful for the patient. And what is the question of the anxiety that you are going to generate? So I think that more and more people are very convinced that, of course, it's interesting to use that as a template, but once again, you should not have a dogmatic position. And to conclude, and I think that probably the situation is more consensual this year than it was before, because we have the reality, and if we integrate the diversity of the severity of what we called before LFS, that we should call now P53-related cancer, if we integrate the incomplete penetrance, if we are considering the variable penetrance, if we should address the question of psychological impacts of annual tumor screening, starting from the first year of age, it would be ridiculous to have a dogmatic position and to offer that to all the patients. So, of course, we have not reached a perfect knowledge, but today it's clear that we should have no dogmatic position like in personalized medicine. The idea is to use the Toronto protocol as a template, but probably to consider case by case, family by family, in order to be very helpful and to guarantee a quality of life, to propose what we call a stratified monitoring, carefully discussed through multiple disciplinary sessions, and taking care today on 3 parameters. Is there a familial history? It's very important because you have childhood tumors for parents that are going to ask to start the follow-up very early. And as you know, familial history means something, probably genetic factors. And maybe in 1 year from now, we are going to identify more and more modifier genetic factors. Type of alteration, one part, you are not going to perform the same screening in only late onset cancer characterized by breast cancer. You are not going to start obviously very early the screening. And as I mentioned, the question of the previous cancer is absolutely critical because we know that if you treat a patient by chemotherapy and radiotherapy, he will have a high risk of multiple primary cancer. Is the reason why it will be very important to offer at least annual whole body MRI. So I will stop here. I would like to thank all my collaborators, my own colleagues who are working very hard for the patient benefit like in other groups. And it was, of course, a national study and now an international study. And now we try to share and to combine the expertise at the European level in the context of the European Reference Network. So thank you for your attention.
Thank you very much. So we have 5 minutes for questions. So please go to the microphone if anybody wants to ask something. Well, I just want to start with one question. And we go to gene panels. And do you think it's problematic to have P53 on these gene panels?
Many thanks. I have a bias. I am strongly against. Strongly against, right? I don't claim that I have the truth, but I will tell you, we had this discussion with colleagues in France, and they say, well, people are doing that in Canada, private society, we should do it. The result is the following. Since they are doing that, I'm getting mails every day. Thierry, we identify that. What should we do? Is that a class 3 or class 5? Because, of course, and in the panic in the patients, because they say, for instance, you have a lady having breast cancer at 53. They are going to identify, of course, and classify a class 3. And, is that an LFS? So it's absolutely a panic. And based on the study, even published by the Dana-Farber group, have you seen the mutation detection rate? It's ridiculous. So I think it's ridiculous today. I'm against, but against, I know that the risk today, it's not only for the Li-Fraumeni, but as you know, is to skip out the phenotypic evaluation. And maybe I am an old clinician, but it's absolutely critical if we want to guarantee to give a service, is to, as you know, before considering the test, to inform the patient of the consequence. So putting that on a panel is a disaster because people are doing many tests, but the critical aspect is to spend time on the interpretation of the quality result. I think it's more efficient to spend your money, to spend your time of your team trying to interpret the result, to go deeply in strongly suggestive situation, because, and to give the result fast, because in our department, we try to provide the result within 1 week or 2 weeks when there is a burning question in terms of radiotherapy. Though I am against large, I prefer to work more in a more specific way.
I have a question. So it's a little bit more of a basic science question, but the spectrum of tumors that associate with Li-Fraumeni is quite interesting. Is it understood why other cell types or tissue types don't have a high frequency of malignancy? For example, colon, prostate, lung, etc.
That's a very important question, and Pierre Reynaud, who is coordinating this aspect, published an interesting paper. Probably that's something to do with the need of the PP3 biology at the stem cell level. For instance, we consider that probably some stem cell level from which are originated sarcoma or central nervous systems require a perfect dosage of PP3 during the development. And as you probably know, we know that PP3 occur very late in the evolution, and because PP3 is also required for apoptosis during embryonic development. So probably that's something to do at least if you are considering the typical spectrum observed in the young patients, something to do to the requirement of PP3 at the stem cell level.
I just have one final question. You have a P53 database in France. So is that a good place to go if you want to, if you have a variant? A
database?
Yeah, I saw you have a P53 database.
Well, I mean, it's not, yeah, well, I mean, it was, it's international because the P53 was created a long time ago by Thierry, now Pierre Reynaud and Magali Olivier. So it's international. Because it's sponsored by the International Agency for Cancer Research. And the idea is to summarize all the somatic plus germline. But you're right, we try now to update and to update not only the germline iteration, but all the characteristic in terms of biological activity.